أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم ألم نخلقكم من ماء مهين فجعلناه في قرار مكين إلى قدر معلوم فقدرنا فنعم القادرون ويل يومئذ للمكذبين صدق الله العلي العظيم. Lecture number 3 for the first year class, and translocation is the topic. Growth is the topic, with which I will discuss two types of the growth. One is a primary growth, one is a secondary growth. Meristems I will discuss apical meristematic tissue, lateral meristematic tissue. I will discuss the opening and closing of the stomata, and especially the flowering mean vernalization. So I start from translocation. You can see the figure. You can see the food molecules can move through a phloem in the form of the sucrose. So I will label that. You can see, first of all, I would like to define plasmodesmata. What is the plasmodesmata? That is the single openings present in the cell walls. Those openings are called plasmodesmata. So you can see the plasmodesmata. These are the plasmodesmata. Food is produced in photosynthetic part, and what happened, where the food is produced, that area is the source. Where the food is not produced, that area is the sink. So food always flow from the source to the sink. So that is the way by which the translocation take place. And the cell which is the present in the phloem, which transport the food, that is the phloem tube cell. And phloem tube cell is helped by a small cell which help phloem tube cell, that is the companion cell. I am drawing figure of companion cell. You can see companion cell has the nucleus. This is the nucleus, and the tube cell is without nucleus. So what will happen? Companion cell will help phloem tube cell to move the food molecules from the source to the sink. So this is the way by which the translocation take place. So it means that companion cell of the phloem tube cell will help in the transport of the food in the form of the sucrose. So what will happen? Once again I will tell you, you can see I am just rubbing some colors, and I would like to show the method of transport of the sucrose. Yes, this is the photosynthetic part. And this will produce the food goes to phloem tube cell, and each phloem tube cell has small pores in the cross walls. These small pores are called sieve plates. These are the small pores, therefore they create the pressure, the movement of the sucrose from the high concentration to the low concentration, mean from sucrose to the sink. That is additional factor to enhance the speed of flowing of the sucrose from the source to the sink. And by the plasmodesmata, mean openings in the cell, it will enter into the sink, mean where the food is required, that area is the sink. So now we talk about, this is the translocation. Now we talk about some other factors which are very important in case of, this is the plasmodesmata. I told you there are three pathways by which water is transported. One is apoplast pathway, which is these are the factors which affect the rate of transpiration. And light increase rate of transpiration, temperature increase rate of transpiration, and of course these are the factors. Wind also increase the rate of transpiration. Transpiration is of three type. One is a stomatal transpiration. Stomatal means that take place by the stomata. One is take place by lenticels. Lenticels are the pores in the stem, and that transpiration is a lenticular transpiration. And the third one is the cuticular transpiration, which take place through the above layer, the layer which is present above the upper epidermis. That layer is the cuticle, and above the upper epidermis, cuticle layer is present. Some water molecule goes into vapor through the cuticle layer also. That is called as cuticular transpiration. Transpiration definition I did in the classroom as well, that movement of the water molecule in the form of the vapor. This is the cross section of leaf. You can see xylem, phloem, upper epidermis, and this is the lower epidermis. I will talk about opening and closing of the stomata. That is the guard cells. Here is the guard cells. The guard cells, how they works, their outer surface is thin, inner surface is thick. So when they become turgid, the opening will develop between them. that is the stoma or stomata, by which, now this is the question mark, why stomata open, how the guard cells become turgid. There are two hypotheses which I will discuss here. One is a starch sugar hypothesis. Starch sugar hypothesis mean this is the guard cell. So what is happening? The guard cells are the only cells in the lower epidermis which are photosynthetic. They will produce the food, and food will be stored in the guard cells in the form of the starch or sucrose. Solute more, solvent less. Solution is inside hypertonic. Hypertonic mean solute more, solvent less. So what will happen? The water molecules will move from outside to inside, from outside to inside, and the cell become turgid, and thicker layer will not move much, but these thinner layers, the end walls of the guard cell, technically concave side will be thick, and this upper side, lower side will be thin, and these openings will be closed due to turgidity. These openings will be closed due to the turgidity, but this opening will not be closed, and the opening will develop, that is the stomata, when the guard cells become turgid. This is the first hypothesis, that the guard cells become turgid. Now I talk about the next factor, that how the influx of the this is very important factor. Light, you know, is very important in photosynthesis. The light will move potassium ions, from outside to inside, So what will happen? Who will move the potassium ion from outside to inside? Dear students, the blue light, which is present in the 7 wavelengths of the light, the blue light is very important. It will move the potassium ions into the guard cells. Same factor will occur. Potassium ion more, it means solute more, solvent less. Solution becomes hypertonic, and the water will move from outside to inside, and the guard cells become turgid, and due to which what will happen, that the stomata openings will be there, and the guard cells become turgid stomata, and those openings will allow some gases to move inside and outside. Exchange of the gases takes place through the stomata. So we go back to next one, which one is a two type of the growth. I will tell you, stomatal transpiration. I have already discussed types of transpiration, and that is not much included in the syllabus as well, because we should go to that topic which is more important according to stomatal transpiration. By the stomata I will speak, no problem. Now I will speak about the cohesion tension theory. What is the cohesion tension theory? Cohesion tension theory explains how the xylem absorb water and how the transpiration help ascent of sap. Ascent of sap and cohesion tension theory almost same thing. Look here. This is the xylem, and xylem consists of the tube which move phloem transport water food, as I discussed, because companion cell and phloem tube cell. But in case of the xylem tube transport water from the roots by 3 pathway, which I will discuss: apoplast pathway by cell wall, symplast pathway by cell membrane, and the vacuolar pathway by the vacuole. So there are 3 main pathways by which the water molecules can reach the root. Okay. And now I talk about, and plasmodesmata is the opening between the cells by which the water can move out during the symplast pathway. So when I talk about the water, these are the water molecules. I make water molecule, I give some colors so that you can, this is a water molecule, this is a water molecule, blue color water molecules. So this is a leaf which is the present at the aerial part, the upper part of the plant. So water molecules which comes out from the leaf in the form of the vapor, that is the transpiration. So when the one blue molecule, mean one water molecule, will move from the leaf, remaining molecules will move up like this one. So why they will move up? Because the space will be created. So this is called ascent of sap. Sap mean water plus solute. Why I am talking about solute? Because water is not alone. Some molecules are dissolved, minerals are dissolved which are required by the plants, like potassium ions, like sodium ions, like phosphate ion, NPK fertilizers, nitrogen also, nitrates are soluble in water, which is very required for the plant for the making of the protein. So these are the sap. So sap moves up due to the transpiration. This is called ascent of sap. Some other factors which help in the movement of the sap up. That factors are, now I am explaining cohesion tension theory. Cohesion tension theory. What are the factors which help in movement of the sap up? One is a transpiration, which I told you. Now I speak the attachment of the water molecule with each other. You know water molecules attach with each other by hydrogen bondings. Yes, of course. One water molecule, this is another water molecule. They are attached with each other by the hydrogen bonding. They are attached with each other by the hydrogen bonding. Hydrogen bonding, you know, oxygen is a electronegative atom and hydrogen is a electropositive atom. Due to hydrogen bonding, these water molecules attach with each other, and due to which what will happen when they will move up in xylem tube, when they will move up in the xylem tube, they will be just like in a line, just like they are going in the lines. So one water molecule move, other molecule also move. This molecule move up, this molecule also move. Reason? Because they are attached with each other by hydrogen bonding. Hydrogen bonding. This attraction of the water molecules with the water molecule is called cohesion. So the cohesion is very important. Now I will tell you what is the water chain. Look here, this is very important. When you want to move this molecule from the xylem, when you want to move this molecule from the xylem, what will happen? The next molecule automatically will move up. Reason? And third molecule automatically move up. Fourth molecule automatically move up. Reason? Because they are in the form of the chain due to hydrogen bonding. That is the tension. So due to tension, the sap moves up. Cohesion, force of attraction between water molecule helps in the movement of the sap up. The third factor is here is not written, but I will explain, adhesion. Third factor is adhesion. What is the adhesion? Adhesion, I tell you. Look here, this is the wall of the stem, this is the wall of the xylem, and this is the wall of the xylem. These are the water molecules. The force of xylem composed of lignin, you know, of course cellulose, but lignin also. So lignin and water molecule force of attraction. Lignin and water molecule force of attraction mean force of attraction between two mediums. One is a liquid medium, one is a solid medium, that is the adhesion. So what will happen when you are moving the meniscus up? Look here, this is the meniscus. Meniscus mean the sides wall of the water. Look, sides wall of the water is up. Reason? Because this is the force of attraction between water and xylem wall. This is the force of attraction between xylem and water wall. It is moving up. Two forces acting up. One force is acting down, means central water will no have xylem, but net force will be up and sap will move up due to adhesion. So this is called as cohesion tension theory. Now I talk about, dear students, I will talk about some other factors which what is the primary growth and what is the secondary growth. But before that, I would like to explain the apical and the lateral meristematic tissue. At the top of the stem, this is the top of the stem, there are some cells which can do mitosis, which can form new cells. So all new cells are formed by the top of the stem only, because these cells are only allocated to divide to form the new cells, and these are called apical meristematic tissue. And some cells inside the stem here, they increase the thickness of the stem, and these are called secondary meristematic tissue, or you can say lateral meristematic tissue. By apical meristematic tissue, the stem increase in the length. This is called as primary growth. And by the lateral meristematic tissue, mean the meristematic tissue which is the lateral meristematic tissue, the meristematic tissue which is present inside the xylem and phloem in the center. This is called as lateral meristematic tissue. It increases the thickness of the stem. So thickness of the stem will be due to lateral meristematic tissue, and they will be called as secondary growth. When thickness increases, that is called as secondary growth. Now I would like to tell you what is the secondary growth. Once again, there is a xylem. This is a xylem. This is a I will make a layer. This is called cambium. In the cambium, This is called vascular cambium. It is present in the stem. Okay, so I am making cross section of the stem. Vascular cambium is meristematic tissue, lateral meristematic tissue. One is the tissue which I told at the top of the stem called apical meristematic tissue. One is the tissue which is present at the stem, which I am discussing. Cambium, vascular cambium. It will form secondary xylem inside. These are the rings of xylem which I am making inside, and it will form outside phloem, secondary phloem. You are understanding? So this is the main tissue which I am naming. I should make color, yeah, different here. This is good. This is the color. I should use any other color for that. This is the vascular cambium. This is the vascular cambium. They are forming secondary xylems inside, and secondary phloems will be outside, and secondary xylem increase the thickness because they will be formed more. More rings are produced by vascular cambium than the secondary phloem. These more rings mean thickness more. This is also called wood. In other words, secondary xylem is also called wood. It is also called annual rings because each ring of secondary xylem produced in 1 year, so that age of the tree can be also calculated. So this is the primary and secondary growth. Now I would like to talk about some other things which I want to discuss with you. For example, I would like to tell you about the transport. Yes, I would like to tell you about the vernalization. What is the vernalization? There are two mechanisms by which flower is induced. One mechanism is photoperiodism and one is a vernalization. Photoperiodism, one is a photoperiodism, one is a vernalization. What is a vernalization? That is flowering takes place in the plant. That is a vernalization. And vernalization takes place at 4 Celsius. At 4 Celsius. Okay. So I will tell you flowers. What are these two processes? The flowering takes place due to mechanisms. One is a vernalization, one is a photoperiodism. What is a vernalization? Vernalization means that flowers are induced at 4 Celsius. Mean you will give to plants, you will keep plant experimentally you will keep the plant at 4 Celsius. Flowers will be introduced. Which temperature? 4 Celsius. But if you will in case of photoperiodism, this is vernalization. So what is the photoperiodism? That is not in the syllabus. That is quite a lengthy topic, but I will speak only two things in reference to photoperiodism. There are some phytochromes, which are the pigments present in the plants, and they are of two types, and they will absorb the different wavelengths at the different time. Mean at night time they will absorb different wavelengths, at the daytime different wavelengths. And there is one hormone also, florigin, florigen, florigin hormone, that also causes the flowering. So when you are increasing the number of the flowers, or you are studying flowers in reference to phytochromes, different type of the phytochromes, which type of light they are absorbing, and the hormone like florigin, and that's mean you are studying photoperiodism. Once again I will go to vernalization. When you are introducing the flowering by the temperature 4 Celsius, you are giving a significant temperature, you are maintaining the significant temperature for the flowering, that is the 4 Celsius. The flowers will be induced more, and this process is called vernalization. So vernalization is related with the induction of the flower by a temperature based phenomena, that 4 Celsius. And photoperiodism also flower induction, study of flower formation by pigment like phytochromes and florigin. So dear students, we discussed the translocation, apical meristematic tissue, lateral meristematic tissue, cohesion tension theory, and stomata, opening and closing of the stomata, and we studied also vernalization. And this was lecture number 3. Thank you very much.